Welcome to the fascinating world of evolutionary biology. Today, we're going to explore the diversity of chordates, specifically focusing on the echinoderm theory. We will uncover a mind-blowing secret about our deep ancestry and discover why your distant evolutionary cousin might just be a starfish. Let us dive in. Now that we've introduced this surprising topic, imagine looking at a massive family tree and finding out that your distant cousin is a starfish. It sounds absolutely crazy, right? A starfish looks nothing like a fish, a dog, or a human being. But in biology, adult looks can be highly deceiving. If we only look at fully grown bodies, we miss the bigger picture. To find the truth about our origins, we have to look at how a creature is built from the very first cell. This is why scientists turned their attention to microscopic embryos. Building on that idea of looking at embryos, we arrive at the formal definition of the echinoderm theory. This theory proposes that chordates, which includes every animal with a spinal cord, and echinoderms share a direct common ancestor. Because adult forms are so different, this evolutionary theory is primarily based on embryological, larval, and biochemical evidence rather than comparative adult anatomy. Essentially, the theory states that our earliest chordate ancestors diverged from a primitive, free-swimming larval form of an ancient echinoderm. It is a profound shift in how we view our lineage. To understand why embryos are so important here, we must look at a fundamental split in early development. In both chordates and echinoderms, during early embryonic development, the very first opening that forms in the embryo becomes the anus. This specific condition is known as deuterostomy. In stark contrast, most other invertebrates are protostomes, meaning their first embryonic opening becomes the mouth. Because both we and starfish develop anus first, it provides massive evidence that we share a deep, common developmental pathway that other invertebrates simply do not have. Beyond the first embryonic openings we just discussed, the theory also highlights striking similarities in later larval stages. Scientists noted a strong morphological resemblance between the hypothetical Diplurula larva of echinoderms and the Tornaria larva of hemichordates. These tiny, free-swimming, bilaterally symmetrical larvae act as an evolutionary bridge connecting simple sea creatures to true chordates. By comparing these specific larval forms, biologists can trace a clear, step-by-step physical transformation that eventually led to the complex spinal cords we see in modern vertebrates today. This larval bridge leads us to a profound biological principle called paidomorphosis. Understanding the echinoderm theory is absolutely crucial because it solves the mystery of where vertebrates actually came from. Without it, the sudden appearance of complex chordates in the fossil record seems impossible. Paidomorphosis shows that evolution often repurposes early developmental larval stages to create entirely new species, rather than modifying highly specialized adult forms. In the real world, this concept guides geneticists and medical researchers who study human developmental diseases by looking at the basic genetic pathways we still share with simple marine organisms. Now that we understand the biological concepts, let us look at how scientists quantify these relationships using mathematics. While evolutionary biology is heavily based on observation, cladists use a formula known as the Trait Similarity Index to turn biological traits into hard numbers. In this formula, I represents the Trait Similarity Index, expressed as a percentage. S represents the number of shared developmental traits between two groups. U represents the number of unique traits found only in one group. When the number of shared traits increases, the index gets closer to 100%, indicating a strong evolutionary link. Conversely, if unique traits increase, the index drops. Before we move on, let us see this formula in action with a concrete example. Suppose a biologist examines 10 fundamental embryonic traits to test the echinoderm theory. They find that echinoderms and chordates share exactly 8 traits, such as radial cleavage, so S equals 8. However, chordates develop 2 unique traits, like a notochord, so U equals 2. We substitute 8 for S and 2 for U into our equation. The denominator becomes 10, representing the total traits. Dividing 8 by 10 gives 0.8, which we multiply by 100. The final answer is 80%, showing a massive evolutionary overlap. Now that we have calculated the Echinoderm index, let us compare chordates to a completely different group, like annelids or earthworms. Out of 12 fundamental traits examined, the biologist finds they share only 3 basic traits, like bilateral symmetry, so S equals 3. They possess 9 unique traits that drastically differ, so U equals 9. We plug these new values into our formula. The numerator is 3, and the denominator becomes the sum of 3 and 9, which is 12. Dividing 3 by 12 yields 0.25. Multiplying by 100 gives a final index of 25%. So, we have successfully calculated both indices. This brings us to the logical proof of our theory. Let us evaluate what these numbers actually mean for evolution. In our first calculation, the index for echinoderms and chordates was 80%. Physically and practically, this exceptionally high percentage strongly supports the echinoderm theory. It proves mathematically that despite their vastly different adult forms, these two groups are deeply related at the embryonic level. This high score is exactly why scientists confidently link our ancestry to ancient starfish relatives, setting a strong baseline for our evolutionary tree. Continuing our logical derivation, we now compare our baseline to the alternative hypothesis. The index between chordates and annelids evaluated to a mere 25%. Because 25% is significantly lower than the 80% we calculated with echinoderms, this mathematically proves a crucial biological point. It explains exactly why scientists reject the older idea that vertebrates evolved from worms or insects. The numbers force us to accept the echinoderm theory as the most logical and scientifically sound explanation for our origins. We have proven that the math perfectly matches the biological observations. Now that we have proven the theory mathematically, let us visualize this connection. Here, you are looking at the larval evolutionary bridge diagram. On the far left, we see the Dipleurula larva, representing our ancient echinoderm ancestor, complete with its mouth and anus-first development. Following the arrow to the center, we see the Tornaria larva of hemichordates, which looks incredibly similar in structure. Finally, the arrow on the right leads to the primitive chordate larva, which now features a dorsal nerve cord and a notochord. This diagram beautifully illustrates the step-by-step physical transformation across these ancient microscopic stages. To wrap up our lecture, let us review the most important ideas we have covered. First, chordates and echinoderms share a direct, common deuterostome ancestor, meaning we both develop anus first. Second, we learned that the deepest secrets of evolution are often hidden in the earliest embryonic and larval stages, not in adult forms. Third, we used the Trait Similarity Index to mathematically prove that our developmental overlap with echinoderms is incredibly high compared to other groups. Ultimately, this theory reminds us that the grand tree of life connects the most unexpected creatures. You now fully understand the echinoderm theory.